Hi everyone, my name is Vrinda and I'll be presenting some of the work that I did during my PhD at the National Center for Biological Sciences in Bangalore, India. Over their lifetime, all organisms can encounter incredible changes in their natural environment. So, for instance, here you can see a patchy grassland landscape where topography, soil, and vegetation show significant heterogeneity within relatively short distances. Now, animals have to navigate such heterogeneous landscapes and make decisions that impact their survival and reproduction all the time. In this talk, I will discuss oviposition behavior in heterogeneous environments and how that impacts maternal fitness. Studies done so far document a lot of unexplained variation in oviposition behavior, and often this behavior is thought to be suboptimal because it doesn't reflect the best oviposition preference. However, in this talk, I will be showing you some recent data that hopefully convinces you that analyzing reproductive behavior and its fitness consequences through the lens of context dependence can entirely change our conclusions about how adaptive it is. While it is probably obvious to many in this audience that all behavior is context dependent, I would like to show you how we demonstrated it using a fitness landscape framework. So, what are the contexts that I used for this study? I use age and density, two contexts that commonly vary for most species and are fairly universal. We already know that both of these often lead to a reduction in individual fecundity for many species. And even the model organism that I use for the following experiments show low fecundity when female age and experience density increases. I did the following experiments with the red flower beetle, shown in cartoon form over here. This is a pest that infests storage grain and flour warehouses. And while it's difficult to know for sure, we suspect that individual females of this species experience a lot of resource heterogeneity in these warehouses. Female beetles also reproduce through much of their lifespan and have to constantly make reproductive decisions in these heterogeneous environments. So, to put all of this together, I want to ask whether female beetles that have experienced different age and density contexts also show different behaviors while ovipositing in a heterogeneous environment. Depending on whether they allocate more eggs to the optimal and ancestral wheat patch or more to the suboptimal finger millet patch, what are the fitness outcomes of that context-specific behavior? So first we had to establish the fitness consequences for different oviposition strategies. How did we do that? We allowed individual females to oviposit in wheat flour, their ancestral resource. Then we carefully sifted out these eggs and experimentally created different levels of female egg allocation. We did this experimentally by distributing different proportions of eggs from a single female in this heterogeneous habitat that you see here to the top right. We either placed all of a female's eggs in wheat or all of them in finger millet. We also sampled the quartile values of egg allocation. So we had an experimental setup that tested the entire distribution of possible egg allocation in quartiles and simultaneously paired these manipulated egg allocation values with the natural range of fecundity in the species. Using these data, we allowed the offspring to develop for 21 days and measured their survival, development rate, and the total number of offspring. And I will show you what these data look like in the next slide. On the y-axis, we have the percent egg allocation in finger millet, with the rest of the eggs going into wheat in the heterogeneous habitat that I showed you in the last slide. On the x-axis here, we have individual female fecundity, which hopefully also captures some part of the range of fecundity found under natural conditions. Here you can see the data from the experiment I just described to you. Each point on this plot is the data from an individual tested female. The data points are colored by a component of fitness of the offspring, in this case offspring development rate, where yellow represents higher development rates and blue represents lower development rates. I also did this for the total number of offspring, which you can see on the right here. I did not find that offspring survival was impacted at all by egg allocation, so I don't discuss it in this talk any further, but I'm happy to take questions later by email if any of you are interested. Here you can see a smoothened version of the scatter plot I just showed you. And just to reorient you again, here yellow indicates high fitness and dark blue is low fitness for each of these two components. So here we can see that the best strategy to maximize development rate is to lay very few eggs and to allocate all of them to the optimal wheat resource. This is also the worst strategy to maximize the total number of offspring. And what you see reflected here is actually a very well-known fact about flower beetles. The total number of offspring and individual offspring development rate often trade off with each other in this species. So if you want to maximize development rate, you have to do so at the cost of the total number of offspring. Now, the next question I asked was whether females from different contexts make decisions that maximize these different components. What we have here is a baseline of what the fitness components are for a given combination of fecundity and egg allocation. Now, I wanted to ask what are the actual oviposition strategies of females from different contexts? Do they vary in a way that give us hints about the context-specific selection pressures that they may be under? So to do this, I generated females from different context groups for both age and experience density. So for instance, I had two groups of young females, one which was isolated prior to the oviposition test and another which experienced high density. I also had three more groups for old females, two groups that were aged under either isolated or high density conditions throughout their lives, and another which was switched from high density to isolated conditions to age till that group was old as a sort of intermediate treatment. After creating these context groups, I tested their actual oviposition preference in the same habitat I used earlier to generate the fitness landscape. I have superimposed the median fecundity and preference of these two context groups here on both the landscapes, so you can see which of the fitness parameters end up being maximized. Here we see, for instance, that within young females, the ones that experienced low density conditions oviposited in a manner that maximized the total number of offspring at the cost of offspring development rate. However, young females that experienced high density oviposited in a manner that did the opposite. In general, it seems that with increasing age and density, females oviposit in a manner that maximizes offspring development rate. I don't have the time to go through each of these treatment groups shown here, but I can take questions over email or Twitter. In summary, we find that females oviposit in a manner that maximizes different fitness components depending on their internal and external context. So for instance, it is possible that under high density conditions where competition faced by a female's offspring is intense, it may be beneficial to distribute eggs such that their development rate is maximized. On the other hand, under isolated conditions, females may have the luxury of ovipositing in a manner that maximizes the number of her offspring, even at the cost of their development rate. After having raised these intriguing possibilities, I want to highlight that we do not actually know if these selection pressures led to the context-specific behavior that we observe here, but it's just an interesting future direction to pursue. I want to thank Deepa Agashe, my PhD supervisor, Gaurav, and the Adaptation Lab at NCBS for their help and feedback. You can find my contact information here if you have any questions or suggestions, and you can find the preprint of this manuscript pinned to my Twitter profile. Thank you so much for listening, and I hope you enjoyed the rest of the conference.